All right. So, all right. Yeah. So this is the latest data set I've been uh, trying to import into my visual analytics platform. It's from BraTS competition. Let's see. What do we know about the BraTS competition? I think I sent a link to it on our Facebook chat.
You went to Australia for that, no?
No, that was a little different. That was a little later.
No.
No, that was I wasn't involved in the BraTS competition. This a BraTS
Are you
comp.
pres-
What?
are you presenting anything? I cannot see anything.
Oh, really?
Just the your image.
Really? Okay. Yeah, okay. Well, let's fix that. Really? Stop. You stopped sharing your. Says I'm sharing my screen. Let's try sharing it again. What? Share screen. Entire screen right here. Also share system audio. Okay. How's this? Is this better?
Yeah,
You see it?
it's coming now.
Oh, cool. Okay. Okay. So, yeah. I think I had sent Amira, well, yesterday during our discussion. I sent, I posted right here a link to the competition that I got this. It was just it's a competition to analyze MRI data. There it is, and segment tumors to segment out the tumors from the healthy cells. So it's so yeah, there's like a few dozen patients with brain cancer, glioma. They went and got MRIs, and then segmentation, pediatric tumor, brain metastases, different brain tumors. So you know, so basically, they had some professional radiologists look at the MRIs and kind of circle the tumors, and then they had a bunch of coders write AI code to do that, and then they compared, the AI results to the gold standard. So so that's the data set there. So I imported it, and I imported the segmentation. So the segmentation is basically just ones and zeros. So it's like a 3D matrix, and it has a 0 for regular brain tissue and a 1 for a brain tumor voxel. And so I imported that data in here, and so there's a big and so I represent the regular brain tissue with cubes like these. So these are, and this is just a random sampling from brain tissue. And then this is the full data set for the tumors. So this patient has a big tumor right here and doesn't really have any other tumors. And you can use my software. It's got two windows, so you can compare two different tumors. So this is another data set. This is 10017. This is 10016. And so this and I can use you can use these sliders to adjust the size of the cubes so that they get smaller, and you can focus on the spheres. So these are the tumors, and I can adjust the color here. So this is a clustering coloring. So the healthy tissue is white or kind of light purple, I guess, whatever color it is. The tumors, different colors here. This patient has a lot of tumors all over the brain. See, you got a little tumor here, a little tumor there, a little tumor there. This patient only has one big tumor right there. So you can use this to compare two different um, MRI data sets in 3D. But what's interesting, what I do here is I apply the Tumor Connectome Framework. And so I got this off of GitHub. Let's see, GitHub TCF. Let's see if it'll find it. Uh, that's different stuff there. Let's try Tumor Connectome. Yeah, yeah, we got lots of regular connectomes. No, but this is the paper, and this is the paper that I'm that I've been inspired to do this from. So, mapping intratumoral complexity interaction networks using machine learning by Vishwa Porak et al. So they basically do radiomics. It's a radiomics platform that uh, analyzes different different voxel parameters in a multi-parametric uh, MRI data set, for example, that contains different weighting images. So it'll have like T1, T2, and maybe flare. It has all those types of multi-parametric data sets. And so this is how they visualize it. So they take a slice and there's a tumor, and then they do connectomics on it, and then colorize uh, the uh, the graph theory metrics and identify different tissue types from that. So so um, the visualization. So they calculate geodesic distances. And they do it in 1D, and I'm doing it in 3D. So they've got they've got a multivariate patient classification. So they use an SVM to so they have the patients.
So, may I
Yeah,
ask you a question?
sure.
Tumor connectomes, what molecules it connects?
What does it connect?
Yeah,
What's your question about it? The connectivity?
the connectomes are with what sort of molecules, proteins, or anything?
Well, this is well. So um, it's it doesn't connect to proteins or molecules, but well, the MRI has different pulse. So MRI works by you have a strong magnetic field, and it aligns all of the atoms so that they're all spinning in parallel to each other, and then it blasts radio frequencies. It scans through the brain with or whatever other tissue it's imaging with a beam of radio waves. It scans every voxel. It goes left, right, up, down, in, out, and it just scans the whole brain. That's why it takes an hour. It gives each voxel a pulse of radio or a series of pulses of radio waves, and it listens for echoes. And then it moves on to the next voxel and the next voxel, and it does this for the whole volume of the brain and creates a 3D image.
Oh,
Um,
cool.
so and it does and it you can vary the pulses of radio waves so that you get different kinds. It's basically nuclear magnetic resonance. And NMR is used, in a chemistry to identify proteins and molecules based on different peaks at different frequencies. This is not quite as fine grained, but it still gives different uh, different echo readings from different pulses for different types of tissue. So the tissue, you can tell what kinds of cell types are in different tissues. So in the brain, this is a whole bunch of, so they got flare, T2 flare, T1 MP rage. And they have, these are, the different pulsing parameters here. And then they did diffusion weighted imaging. My data set didn't have that. So, and these
So
are,
what is the actually connect? I mean, what are
uh,
the
well, the connectomics is just kind of an analysis method where you just take every voxel of the tumor and you mathematically collect the various dimensions of data for each voxel and compare it to every other voxel for similarity. So the theory is that, similar, if two voxels have similar readings for T1, T2, and flare MRI types, then they should have the same distribution of cancer cells. So glioma is a heterogeneous tumor. There's the cancers differentiate into different types of cancer cells, and the way they like invade and infiltrate the surrounding tissue, the degree of infiltration and the you can, we're trying to kind of understand the cell type distribution in the tumor by comparing uh, the the uh, the cell types in each region of the tumor, in each 3D region of the tumor. So yeah, like so you got post contrast degrees. So and they're using, and so once they have a connection, a connectivity, um, a connect. So there, brain tumor connectomics. So they had 24 patients, and let's see, ADC segmentations, peritumoral edema. Yeah, and so there's different kinds of cancer cells. There's different kinds of immune cells in and so we're trying to get an idea of, the mixture of different cells in each area of the tumor microenvironment. And so some of the conclusions that they drew here is they had grade 4 tumors demonstrated greater betweenness centrality and average path length than grade 2 tumors. So this is what they concluded from their study. So they compared every tumor region with every other tumor region, found the similar and create a similarity or difference, if you want to have to have the glass half full or half empty, between all the different microenvironments in the tumor, and they calculated the betweenness centrality and the average path length, and they found differences, significant differences between grade 4 tumors and grade 2 tumors. So this is another way of classifying a brain tumor. And
okay?
so, we can,
So
look
do
up.
they compare
Yeah.
normal brain cells with the?
I I don't know. I don't think so. Yeah,
Okay,
probably. I mean, I'm sure they do somewhere. So but yeah. Well, no, not really, because normal brain cells are, they're not, they're not gonna. I mean, that could be something we could work in. I don't know if they can work that in. I mean, we could work that into our equation. I mean, that's something we can do, I suppose. I don't know if that'll be useful. Um, but then here we'll look up betweenness centrality. So betweenness centrality in graph theory, betweenness centrality is a measure of centrality in a graph based on the shortest paths. So every pair of vertices is in a connected graph. There exists at least one shortest path between the vertices. So um, such that each number of edges that passes through for unweighted graphs or the sum of the weights is minimized. The betweenness centrality for each vertex is the number of these shortest paths that pass through the vertex. So for each vertex, the number of shortest paths between every vertex and every other vertex is its betweenness centrality. So they calculated the betweenness centrality uh, of each volume of space in a tumor and compared it to grade between grade 2 tumors and grade 4 tumors and noticed a difference. And they also did average path length. So I'll look up what that means. Average graph path length. Well, this is some kind of a math homework problem there. Wikipedia. So the average path length or average shortest path length is a concept of network topology that's defined as an average number of steps along the shortest paths for all possible pairs of network nodes. So the measure so I guess what they did was like they calculated so for each voxel they in the tumor they calculated the path between it and every other voxel or the shortest path and and then they took the average length of all those and assigned that number to it. So then they do that to all the voxels as the central voxel, then that's how they come up with the shortest path centrality. And so they're trying to and then they so that's how they came up with like these images like here. Let's see, how do I zoom in on this? So there's the degree centrality and the average path centrality. So they calculated the degree centrality between every voxel. Well, I guess the betweenness. This isn't betweenness. This is degree central. That's a different type of centrality. The centrality kind of gives you an idea of how important a node is for every other node. And so for the and with regards to a tumor connectome, you gotta kind of take this kind of understand that a tumor is an evolutionary biology process. So the tumor starts with a single cell that's like the great 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 grandmother of all the other tumor cells. It has a mutation and it starts proliferating and it stops checking its DNA between proliferations. So each generation introduces more mutations, and they try to compete for the resources of the host. And so the host immune system and tries to kill them, and the resource finiteness starves the cells. With the cancer cells that can survive better have more proliferation in the next generation. And so we're trying to kind of understand the tumor's hereditary makeup and the the theoretically the graph of the tumor's of the tumor's connectome will kind of give you some information about that, and you can consider the tumor as a graph, of a lineage tree, as the tumor's lineage tree as a graph on the cellular level, but also in this macroscopic voxel level, where each micro uh, environment of each voxel, it contains a mixture of different cancer cells, and in the predominance of the different cancer cells gives you kind of an idea, kind of lets you characterize that tumor microenvironment voxel as a node, and and construct a graph from between all the voxels, and the similarity matrix, and a graph from the similarity matrix, and gives you kind of an ability to assign centrality metrics for how uh, uh, to how for how to various to how any given region might have played a role in the evolution of the tumor, based on the centrality metrics of that graph in that region. And so that's their theory, and they've been able to um, have some success classifying grade 2 from grade 4 tumors in this way. There's a little chart showing AUCs, and rocks and MCC for the different metrics. And yeah, so yeah, I'm gonna get that's pretty much the theory behind my project. And so I'm using the BraTS MRI data. Here you know, maybe I'll unpack a piece of BraTS data. Um, yeah, I don't think I'm going to get any BraTS data from here. Um, but yeah, so I'm using their data to visualize the tumor connectomes. And so these are two different patients. One has a whole bunch of tumors, like tumor, 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 tumor. This one just has one big tumor. But so, if we wanted to kind of focus on maybe analyzing in depth how this patient with multiple tumors, how their tumors maybe are related to one another. Um, so I I run the tumor connectome. I run this paper's algorithm on the raw data set here, and I get a distance matrix, similarity matrix between each region of space that is segmented to classified as being in a tumor, and I I do the tumor connectome framework on it, creating a distance matrix, and then I cluster it. So right now I'm comparing two different brains with two different tumors, but I can use this split screen view to compare one brain's anatomical positioning of their tumors to a dimensionality reduction view of, see, like the MDS, multidimensional scaling. So this is a multidimensional scaling view of, so this takes all of these tumors and organizes them in a multidimensional scaling algorithm. So this is tumor space. So this would be kind of a similar algorithm that you might use
So
if you
why do you
were,
get three different colors?
well, k-means clustering. So that the
Okay.
coloring is from k-means. So it
Oh,
arranges the tumors this in pretty much a similar algorithm that you might use for like genomics or proteomics, where it takes the different levels of various and interesting markers like proteins and genes, and then it just clusters the data sets, the huge dimensional data sets. So if you did like a proteomics or genomics or transcriptomics study on this, you might get like a few thousand dimensions for each sample point, which in this case is a tumor voxel, and then you'd cluster that 10,000 dimensional data set and dimensionality reduce it to 2 or 3 dimensions using like multidimensional scaling or isomap or t-SNE or whatever, and then you'd colorize it by finding clusters using a clustering algorithm like k-means. And so I did that, but instead of using 10,000 data sets with all the genes and proteins and transcriptomes and metabolites, I didn't have access to any of that stuff. All I had access to was MRI data using T1 weighting, T2 weighting, and FLAIR. And so I had a three-dimensional data set, and I used a three-dimensional MDS dimensionality reduction, multidimensional scaling algorithm to display it in. And so this way you have a three-dimensional data set, and you can, and my interface lets you kind of hover over these cells and it'll highlight and every and so this data set on the left corresponds to the tumor data set on the right. They're colored using the same color code, and every you can explore it in site in NeuroCave by mousing over different regions in your cluster, and it highlights the corresponding tumor regions in the anatomical data set over here. And similarly, like if you're interested in a tumor, in a particular tumor, you can mouse over it and it highlights which cells in the dimensionality reduction view correspond to it. And you can click on the tumors or the tumor regions, and it creates these edges that show which other tumor regions have most similar data to it. And so, like, I clicked there and it highlighted this one over here, and it displayed the edges. And you can use a threshold to kind of limit the edges to just the more stronger connection weights. Click on like maybe this one again. So yeah, so now my cutoff is 0.062 on the weight scale, and it uh, displayed the connections to these other. So you can really get an idea of the graph theory that determines the different centralities, and you can also colorize it. So you can explore, so maybe you can kind of build a graph, so you can click on one of those, one of the neighbors, and get an kind of get an idea of what the neighbor's connected to. You can kind of build your graph there. So you can maybe explore some high centrality nodes and kind of get an idea of what their which other regions they're more similar to in both um, data space over here on the left, which is an MDS multidimensional reduction transformation of the tumor data set, and also how it relates to all the different tumors and their migration patterns in in this metastatic brain data set. So so you can kind of see which nodes are involved in which ways and how they're connected to the and how they proliferate throughout the brain's metastatic process there. So yeah, that's what I've been working on there. You can also colorize it with different color codes. So you can use um, I guess maybe degree centrality as the color code. So now it color coded the cells degree centrality. Well, some of them disappeared here. Maybe eigenvector centrality. Some of these don't work. There, the average path length. That's a good one there. So, so I guess these have a different path lengths. So so you can explore ones that have significant path average path lengths instead rather than the clusters that they belong to. So this basically this 3D tool gives you kind of a 3D way to browse this sort of data set here, and really get an understanding of the difference. So that paper found the difference between grade 2 and grade 4 tumors, and I'm interested in studying exactly what that difference is from a tumor evolutionary standpoint using tools to trace the evolution of tumors. And so I guess the PhD is gonna there's gonna be three aims, and they're all gonna be somehow using a uh, an immersive visual analytics platform to basically compare complicated multi-scale data sets. And so in this use case, my aim is to compare the mesoscale of a human brain with kind of a macro scaled human brain with the mesoscaled voxels in the micro environments of the tumors in those regions.
So what do you say? It's voxel.
What's a voxel? Well, it's the same as a
Hmm.
pixel, but in 3D. So a
Okay, okay.
pixel is a 2D coordinate of a 2D screen, whereas a voxel is a 3D coordinate in a 3D MRI volume. So it picks your screen has maybe like 10 million pixels or something, maybe 4 million. I don't know. If you have like 2,000 wide by 1,000 uh, high, you'll have like 2 million pixels. With 2,000 by 1,000, whereas this
And when
data
when
set.
you said when you
What?
say multi scale, what does it mean?
Well, the different scales are so different scales of data. So you have a
Uh,
brain that's about the size of like two of your fists, and then
uh,
it's been decomposed into about 6 million voxels. This data set is about 256 voxels across, 256 voxels wide, and about 100 voxels uh, deep, and so that makes about 6 million voxels, 256 by 256 by 98 or 100 or 102. So those are the two different scales. You got like a mesoscale and a macro scale, and I have some other data sets that I
is
think
there a correlation between these points and basic molecule or atomic?
well,
Uh,
not in this case, but I'm going to
uh,
have a different use case So, like the thing I went to Australia for, that was a proteomic, so they had like drug interactions and protein interactions. So that was maybe I guess the different scales might be um, so we did have protein data. So that had um, I think the different samples. They weren't necessarily from the same patient, but
I mean, can you relate the tumor connectomes to molecules?
maybe. Well, one thing I'm going to want to do here is because I'm trying to talk to a professor um, that does brain MRI uh, tumor classification, and maybe get her on my committee and get some data sets from her that include the diffusion weighted imaging or diffusion tensor imaging, which can which will give me the ability to connect the healthy brain tissues to each other and how they're wired with neurons. So I'll be able to compare the neuronal brain wiring in a tumor patient and how that wiring rearranges. And so I'll be able to perform graph theory on the neural wiring and the evolutionary trees of the tumors and get an idea of how they interact with each other and have maybe get even better classification and maybe even uh, etiology research. Etiology being why tumors happen and how to make them not happen. So or stop happening. So that's that. And yeah. The thing I talked about in Australia was the visualization of like drug interaction data, and tumor interaction data, and how drug uh, effects on protein. Did I say tumor? I meant proteins. How drug, how the drugs protein interactions affect all that, and so um, and of course drugs act systemically, so that's kind of a systemic scale phenomenon, whereas proteins perform are on a micro scale, a nanoscale really. So those are, well, I don't know. That's kind of hard to draw a conclusion on multi-scale things there. Maybe scale isn't that important on my, for my, but maybe scale's just one aspect, and you gotta find it some other way of linking together my different aims for protein. Kind of bridging the gaps between the mesoscale and the micro scale and the nanoscale. Um, but this comparing drugs and proteins interaction networks is something that this can also be useful for as another aim. And with the data set that I'm also currently exploring is uh, for mm, uh, complexes, for protein complexes. So, and I guess the different, maybe scale's not the right word, but the graphs that I'm comparing on that level are the the MCTS tree, where the algorithm tries to assemble a protein complex out of maybe a couple dozen monomers of that complex, and it tries assembling them in different ways, using AlphaFold to determine which one of them works and which doesn't. So that's uh, that's kind of a different scale thing. So you have the complexes are graphs. They're very small graphs where there's not really any graph theory, but they have different connectivity patterns. Which protein their interaction graphs in the MCTF, the Monte Carlo tree search graph, uh, is what searches through the different protein interaction subgraphs, and tries to identify the ones that have the minimal energy structures. Hold on, I gotta pee.
Can we stop it today and we can continue later on?
Oh, sure, yeah, yeah. It's probably getting late over there. Yeah, of course. Well, yeah, yeah,
Okay.
yeah. That's post any of your questions online. I'll try to answer 'em.
Okay.
All right, cool. We'll see you later,
Thank
Vinay.
you.
Thanks for
Yeah,
joining.
thank you. Thank you.
Bye.
Bye.